So good morning, everyone. This is the last lecture of this mini-semester for the summer course. So we will be talking about the water and soil physiology of animals in their environment. We will talk about the kidneys and excretion, and also we will talk about the different kinds of animals in dry savannas. So starting with the water and soil physiology of animals in their environment, we all know that each type of habitat that poses different challenges, and animals of diverse types that live in a particular habitat have converged on similar mechanisms for meeting the challenges. So for these reasons, we can say that detailed study of animal water soil physiology is organized around habitats. So we take that approach in this chapter. So we will start by focusing on freshwater habitats in part because we have already discussed them in chapter 5, and in part we will because the study of this freshwater fish will help us to set the stage for a better understanding of ocean fish. So starting with these powder blue surgeon fish, that's the Acanthrus leucosternum, that expend energy to keep their body fluids more dilute than seawater. So the major questions that are raised are why they do so and what are the different mechanisms they employ. So since we all know that the body fluid of marine teleost, that's the bony fish, for example, we have the coral reef fish, they are far more dilute than the seawater in which they are swimming. So such fish have blood osmotic pressures of about 300 to 500 milliosmolar. So whereas seawater has an osmotic pressure of approximately 100 milliosmolars. So from this viewpoint of fluid concentration, marine teleost fish are the packets of low salinity fluids that are cruising about within a high salinity environment. So how the exchange of the fluids or the salts take place in the ocean. So because the body fluids of a marine teleost fish are not at equilibrium with the seawater that's surrounding the fish, so passive processes always occur there which tend to alter the composition of the body fluids. So we can say that actually the water tends to move out of the body fluids into the surrounding seawater by osmosis, since we all know. So because the body fluids have a lower osmotic pressure than the seawater. So conversely, we can say that there are certain inorganic ions that are present in the water, ocean water, seawater, that diffuse inwards from the concentrated seawater. So both the outward osmosis of water and the inward diffusion of ions, it tend to concentrate the fish's body fluids. So recognizing these processes, we can see that marine teleost fish must steadily expend energy to maintain their body fluids out of equilibrium with the seawater in which they swim. So let's go in detail about this. So this will give us an idea. So going through this chapter will give us an idea about the study of freshwater fish and will help us to set the stage for a better understanding of the ocean fish. So this table here, it shows you the composition of blood plasma in some freshwater animals. So we all know that the animals that living today in the freshwater, they are descended from ocean living ancestors. So major animal phyla that originated in the oceans and later invaded all other habitats. So seawater was probably somewhat different in its total salinity and salt composition in the early eras of animal evolution than we can see today. So the osmotic pressure of fresh water is typically less than 1%, as high as that of seawater today, and major ions in fresh water are very dilute compared with their concentrations in seawater. You can see here in the table this. So table here shows us the all freshwater animals, they regulate their blood osmotic pressures at levels of hyperosmotic to freshwater and are therefore classified as the hyperosmotic regulations. So table in this figure here shows the blood osmotic pressure of various types of freshwater animals that span an order of magnitude. But even freshwater mussels, which are among the most, you can see here, the freshwater mussels, which are among the most dilute animals on Earth, they have blood that's substantially more concentrated than the freshwater body fluids, as dilute as freshwater seem to be incompatible with life. So the solutes in the blood plasma of freshwater animals are mainly the inorganic ions. For example, the sodium positive chloride negative dominates plasma of uh, freshwater animals. So we can say that this table here gives us an idea of individual or inorganic ions in the blood plasma of freshwater animals. In almost all cases, you can see, for example, freshwater mussels, we have snails, crayfish, mosquito larvae, brown trout, frogs, and river water. So these are all the freshwater animals. So you can see the different ion concentration here in millimoles per liter, the different ion concentration of sodium, potassium, calcium, magnesium chloride, and bicarbonates. And also they have different osmotic pressure in milliosmoles per kg of water. So talking about the water-salt relations in a freshwater animal, we all know that being the hyperosmotic to their surroundings, freshwater animals tend to gain water continuously by osmosis. We already talked about that. And this water gain tends to dilute their body fluids. So we can say that relatively high concentration of ions in their blood actually suggests the net diffusion of ions that tend to be from their blood into the ambient water. So when all the complexity is taken into account, the direction of diffusion of the major ions, such as sodium positive or chloride ions, is as the concentration gradient suggests from the blood into the environmental water. So this loss of major ions by diffusion actually tends, like the osmotic water gain, to dilute the body fluids of a freshwater animals, as you can see here in this picture. So in this figure, you can see, like figure A here, it mentions the freshwater animals such as a crayfish. It faces challenges because of passive water and salt exchange. So numbers are generalized approximately values for the osmotic, and these are found in the blood of a crayfish and the ambient water. So you can see the numbers of the values for the specific species of crayfish under specific study conditions. They are already given in the table here for the crayfish. You can see the numbers here. It has 436 osmotic pressure in milliosmoles per kg of water. And this is the sodium, potassium, calcium, magnesium chloride, and bicarbonate ions. So moving on to the figure B of this slide, you can see that it gives us a summary of all the major processes of water and salt exchange, including the energy requiring processes the animal uses to maintain water-salt balance. So antennal glands, or we can say the green glands, which function as the kidneys of a crayfish, open at the basis of the second antennae. So in a broad sense, we expect a freshwater animal energy costs for osmotic and ionic regulation to depend directly on the animal's rate of passive water gain and passive ion loss. So more rapidly, we can say that water is taken up by osmosis, and more rapidly ions are lost by diffusion. So the more rapidly an animal will need to expend energy to counteract these processes to maintain a normal blood composition. So there are three factors that actually determine the rate of passive exchange of water and ions. So one of them is the magnitude of osmotic and ionic gradients between the blood and ambient water. The second will be the permeability of an animal's outer body covering to water and ions. And the third factor will be the surface area across which exchange is occurring. So here in this figure, you can see that this is a single gill filament of an adult teleost fish that's viewed in two ways that emphasize the two principal gill functions. That's the oxygen uptake and the ion pumping. So how actually this occurs is that we all know that the gills of the teleost fish, although often we have discussed about the gills, they are the breathing organs. In fact, they carry out two major functions that serve homeostasis. So in both freshwater and seawater, the gills, which are the breathing organs, they function both as ion regulatory organs and as gas exchange organs. So gills are the principal sites where sodium and chloride ions are taken up by active transport from freshwater, as we have already discussed. So the two images you see here of a single gill filament in this figure help to emphasize the two major functions of gills in adult teleosts. So figure 20 here, you can see the figure this A in this slide. It actually shows the microscopic structure of the filament, so which consists of a thin principal part shaped somewhat like the blade of a feather that bears many thin folds, the secondary lamellae on its upper and lower surfaces. So blood flows through all these parts. The secondary lamellae greatly increases the surface area across which the oxygen can diffuse inward from the ambient water into the blood. So the second figure here, the picture B, you can see it actually shows the same filaments that are visualized in a way that reveals the presence and location of the gill epithelial proteins. So these are instrumental in the ion transport between the ambient water and blood. So in this specific case, the epithelial cells expressing the ion transport proteins are located in parts of the filament other than the secondary lamellae. So moving on to the next slide, which gives us an idea about the mitochondria-rich cell that's surrounded by pavement cells in the gill epithelium of freshwater teleost fish. So we will now briefly focus on the cellular level of organization. For example, the gill epithelium in fish, which consists principally of two types of cells. One is the MRCs, that's the mitochondrion-rich cells that we'll be talking about. And these are also called as the chloride cells. And the second will be the pavement cells, which is here. So these are the pavement cells and these are the mitochondrion-rich cells, which are also called as the chloride cells. So mitochondrion-rich cells are considered to be the principal sites of active ion transport in the gills. So these are a central focus of research on ion transport in both freshwater and marine fish. So like in this figure, it shows a portion of gill epithelium of a freshwater fish that's visualized by scanning the electron microscopy. So in this electron micrograph, we see a single freshwater type MRC, that's the mitochondrion-rich cells that are surrounded by these pavement cells. So this is the mitochondrion-rich cell, which is surrounded by different kinds of these pavement cells. So regarding the two major functions of gills, oxygen uptake is believed to occur across the pavement cells, which usually occupy more than 90% of this gill epithelium. Since you can see here in this figure, so most of the space is occupied by these pavement cells. So more than 90% of the gill epithelium is actually occupied by these pavement cells. So as for ion uptake, the physiologists would like to know where these chloride ions uptake and sodium ion uptake occur. But the problem of deducing the detailed molecular mechanism of the ion transport and localizing them to particular cells is exceedingly challenging, and probably the localization of pumps to cell types all require more research on that. So current models of these sodium and chloride ion uptake in freshwater fish can be discussed further. So number of the MRCs in the gill epithelium of a fish living in a freshwater is variable and under adaptive conditions or control. So one condition that increases the number of these MRCs, or we can say the chloride cells, is the alkalosis, the excess of blood carbonate, or during the alkalosis, we can say the MRCs, besides becoming more numerous, they also modify their cell proteins. So second and fascinating condition that leads to increased number of these chloride cells in the freshwater fish in life is very soft water. That's water of exceptionally low calcium positive ions. So talking about the cellular acclimation to living in two types of water in the gill epithelium of freshwater fish. So in this figure, you see here the sections of the secondary lamellae in the gills of rainbow trout, which is also called as the Oncorhynchus mycis. So this is viewed under the light microscopy and stained to show the mitochondrion-rich cells. So figure A here, it shows actually the tissue section from a fish that has been living in ordinary fresh water with calcium dipositive ion concentration of 0.4 millimolar per liter. So the figure B here is the trout that's living in very soft fresh water. Or we can say that this is the tissue section from a fish that had been living for 2 weeks in very soft fresh water with calcium dipositive concentration of like 0.05 millimolar per liter. So these are in this slide here, you can see the two kinds of the ocean animals with different blood osmotic pressures, and invertebrate with blood isoosmotic to seawater, and a teleost fish with blood dramatically hypoosmotic to seawater. So animal life in the oceans is far more phylogenetically diverse than that in the other major habitats on Earth. So probably it might be a good part because animals originated in the oceans. So we all know that today all phyla and most classes of animals that we have marine representatives. So many phylogenetic groups of animals, they moved from the oceans to the freshwater and land over the course of evolutionary time. So consequently, whereas some modern marine animals, they have a continuously marine ancestry. So others, they trace their history uh, to forms that occupied other major habitats. So we can say that such a phylogenetic history has sometimes left major imprints on the water salt physiology of modern marine animals. Let's consider an example of octopus and the marine, these teleost fish. That's the seen here. So this is the figure of octopus and this is the marine uh, teleost fish. So let's consider the example of these two. So this marine teleost fish seen swimming in seawater, like you can see in the figure, may look like they would have similar blood osmotic concentrations. So their concentrations actually are very different. So history is believed to be the explanation for this, whereas octopuses probably trace a continuously marine ancestry. So marine teleost fish are likely descended from the freshwater ancestors. So moving on to the next slide, which gives us the composition of the blood plasma or other extracellular body fluids in some marine invertebrates and hagfish. So you can see here that most marine invertebrates, they are isoosmotic or nearly or so to seawater. So included are the marine mollusses exemplified by the octopus that we already talked about. and such other marine animals such as sponges, coelentrates, annelids. We have the echinoderms and most arthropods. For the most part, these animals are products of lines of evolution that never left the sea. So they have always lived in the seawater, and this probably explains why they have simplest possible osmotic relation to seawater. So this table here, you can see the ionic composition of the blood plasma that seems universally to differ in detail from the ionic composition of seawater. So the solutes in the blood plasma of marine invertebrates, they are mostly inorganic ions, and the ion composition of their blood plasma tends to be similar to that of seawater. So a particular ion often proves to be relatively concentrated in some animal species, but relatively dilute in others. For example, we have the magnesium dipositive ions that's relatively high in concentration in the blood plasma of squid loligo, but low in that of crab carcinus. You can see here. So in squids, you have the 55.4 millimoles of magnesium dipositive ions, whereas in crab, you can see here we have just 19.5 millimoles of magnesium dipositive ions. So isoosmotic marine invertebrates maintain the differences in ion composition between their blood plasma and seawater by ionic regulatory processes. So how they do that is these animals are relatively permeable to ions and water. So ions therefore tend to diffuse between their blood and seawater with ease, following their electrochemical gradients. So one process that these animals follow or they commonly use to maintain their blood ionic composition is active uptake of ions from seawater at the body surface or from ingested seawater in the gut. So second common process is the kidney regulation of blood composition. For example, in mollusks and some other groups, the excretory organs, they make a urine that's approximately isoosmotic to the blood plasma. So they alter the urine's ion composition, thereby contributing to this ionic regulation. So we have in this figure here, we will talk about a section of mitochondria-rich cell, which is also called the chloride cell of a marine teleost fish. So before that, let me give you a brief intro about the urine. How the kidneys of marine teleosts typically excrete the urine that's about as concentrated as they can possibly produce. So the fact that osmotic pressure of urine matches that of plasma means that the excretion of urine cannot help the fish with their osmotic regulatory problem. So however, we can see the ionic composition of the urine differs dramatically from that of plasma. And the kidneys are the principal organs that carry out ionic regulation of magnesium, sulfate, and calcium dipositive ions. So we can say that for every millimeter of water that's ingested and absorbed and then excreted as urine, a marine teleost is left with an excess of solutes. Because although the water enters its body by hyperosmotic to its body fluids, the water leaves its body, then it's isoosmotic to its body fluids. So from the viewpoint of osmoregulation, therefore, production of urine by marine teleost fish is an outright liability, and we would expect the fish to limit their volume of urine to the minimum necessary excretion of solutes that are not excreted by other routes. So we all know that the gills of an adult marine teleost assume primary responsibility for excreting excess of major ions like sodium and chloride ions from the blood plasma. So the excretion of these chloride ions is active and is carried out by seawater type mitochondrion-rich cells. So this is a section of the mitochondrion-rich cells that carry out this process of active transport. So these mitochondrion cells are in the gill epithelium. So these cells are often called as chloride cells, like we already discussed in the study of marine teleost, because of these well-established excretion of chloride ions. So in fish, soon after hatching, as we have discussed in our chapter 4, the mitochondria-rich cells are principally found in general integument, but soon the cells become localized to the gills. So although the gill epithelium is believed always to transport sodium positive as well as chloride negative ions out of the blood into the seawater, the excretion of sodium positive occurs by mixed mechanisms. Or we can say the elimination of these chloride ions and sodium positive by the gills of marine teleost fish. It provides our first example of extrarenal salt excretion. So excretion of inorganic ions by structures other than the kidneys. So this process is called as extrarenal salt excretion. So these are the mitochondria-rich cells that have an extensive intracellular tubular system, which are composed of branching tubules that continue with the basal and lateral portions of the cell membrane. And here you can see the mitochondria-rich cells that are dense with mitochondria, accounting for their name, and it's surrounded by these pavement cells. So talking about the water-salt relations in a marine shark. So marine elasmobranch fish, they are hyperosmotic but hypoionic to seawater. So what that means is that marine sharks, skates and rays, they are collectively known as elasmobranch fish. They have evolved a novel solution to the osmotic problems of living in the sea. So their blood concentration of inorganic ions are similar to those of marine teleost fish and well below those in seawater. So however, the osmotic pressure of their blood is slightly higher than that of seawater. We have already seen that in our last table. So ever since the elasmobranch strategy of water salt regulation in the sea was discovered, the biologists have speculated about its possible advantages over the teleost strategy. So until recently, the unusual conclusion was that elasmobranch strategy costs less energy because marine elasmobranchs are able to obtain water by cost-free osmosis, whereas marine teleosts must drink seawater and pump NaCl out of it to get water. So about 15% of the elasmobranch species that are alive today occur in dilute brackish water or in the fresh water. So some of them are permanent residents of fresh water, but most also occur in the ocean. So a well-studied example of the later, that's those that live in the oceans, is the bull shark, or we can say the Corcanimus leucus, because of its devastating attacks on coastal swimmers. So when the elasmobranchs that live in the ocean venture into the dilute waters, they lower their blood urea concentrations, somewhat by decreasing the urea synthesis and retention. So they retain actually the elevated blood urea concentrations. Even though doing so, it promotes their osmotic uptake of excess water in dilute environments. So let's talk about the types of osmotic regulations. So many aquatic animals, they face large changes in the salinity of water. One is the animals such as salmon and eels that migrate long distances between the rivers and the open ocean. And second category is the animals that live near the margins of the continents, along the coastlines. The waters of intermediate salinity, for example, brackish waters, they occur in the estuaries, salt marshes, and other settings. So ocean animals actually venture into the brackish coastal waters. They encounter low salinities than they experience when they are living in the open oceans. So Animals are classified as the osmoconformers or osmoregulators. So osmoconformers, sometimes described as the poikiloosmotic, permit their blood osmotic pressure to match the ambient osmotic pressure. So osmoregulators, sometimes called as homeoosmotic, maintain a relatively constant blood osmotic pressure even as the ambient osmotic pressure rises and falls. So like in this figure, you can see among animals that are osmoregulators, regulation is often limited to certain ranges of ambient osmotic pressure. So thus different categories of regulators are recognized. And one common pattern are called the hyperisoosmotic regulation, which are the species that keeps its blood more concentrated than environmental water at low environmental salinities. And we also have some species that are predominantly freshwater animals, but they venture into the brackish water. So this figure here shows this pattern. So animals exhibit the hyperisoosmotic regulation when they pose mechanisms of hyperosmotic regulation, but lack mechanisms of hypoosmotic regulations. So the figure A here shows the three species of hyperisoosmotic regulation. Such regulations is typical of freshwater animals that enter the brackish water. For example, the carp. It occurs only in many crabs of shores or estuaries. And we have the other condition, that's the hypoosmotic regulators. So this figure here shows 4 species of hypoosmotic regulators. So such regulation occurs in many shore crabs. For example, we have the fiddler crab, we have the yucca regulator, and we have the other crabs. So moving on to the next slide, which gives us an idea about the rate of evaporative water loss of insects that often starts to increase abruptly as a at a transition temperature. So evaporation, which is one of the chief modes by which the terrestrial animals, they lose water. So in this section, we will begin our analysis of the physiological principles of living on land by discussing the evaporation. So we have already talked in our previous chapters, the rate of evaporation through an animal's integument, it depends on the difference in water vapor pressure between the animal body fluids and the air. And it also depends partly on the permeability of the integument water. So lipids, which organize and undergo phase alterations as the temperature changes, as evidenced by observing the ketone lipids such as butter. The researchers have long known that if the temperature of an insect, for example, we have the arachnid, is raised, water permeability increases just slightly after a certain temperature called the transition temperature, and then increases dramatically, as you can see here in this figure. So transition temperature is about 40 degrees centigrade in this case, as low as 25 degrees centigrade in some insects, and as high as like 55. So it's increasing. So the marked increase in permeability at the transition temperature is the consequence of lipid melting. So although the transition temperature is often so high that it would not be experienced by individuals in nature, so this is not always the case. For example, we have the cockroach, that's the Periplaneta americana, which is a common household pest. We have, for example, experiences a marked increase in permeability that started at 25 degrees centigrade until 55 degrees centigrade. So moving on to the next slide, which gives us the lipid composition of the water barrier, which may even differ among the populations of a single species. It gives rise to significant differences between the populations in their physiology of water balance. So As you can see in this table, it gives us an idea about the lipid composition of individual changes as a consequence of acclimatation to different environments. For example, we have the ranid frogs, we have the birds and humans, and this is the resistance that they pose. So this resistance is actually calculated as the inverse of conductance, which is the rate of water loss across the skin. So this conductance is expressed as grams water per centimeter square of skin per second, divided by the driving force, which in this case is the difference in water activity across the skin. So moving on to the next slide, which gives us an idea about the differentiation between population of one species in lipid-mediated protection against evaporative water loss. So you can see here that, so this gives us an idea about the northern population and the southern population of these species. So the graph actually shows the rate of evaporative water loss at 25 degrees centigrade as a function of body weight in live grasshoppers of a single species from two geographically separate populations. That's the northern and southern California. So according to current evidence, we can say that the blue dots here, they represent the northern population and the red dots represent the southern population of these. So these differences, so according to current evidence, these populations exhibit genetically controlled differences in lipid composition of their epicutical layer. So these differences, they contribute to the greater resistance of the southern animals to water loss. So in each population, the rate of water loss is an allometric function of body weight. So these are, the relation is plots as a straight line on this graph. So moving on to next slide, we will talk about the animal's total rate of evaporative water loss that depends on its body size, or we can say the phylogenetic group. So if we sum up an animal's rate of integumentary and respiratory water loss, we get the animal's total rate of evaporative water loss, or we can say EWL. So within sets of phylogenetically related species, the broad statistical trend is for the total rate of this evaporative water loss, which is measured under particular conditions to vary with the body size. So important pattern is illustrated in this figure here in the graph using the bird as an example. Small-bodied species tend to have higher weight specific rates of evaporative water loss than related large-bodied species. So there might be two reasons why the small-body species tend to have relatively high weight specific rate of this evaporative water loss. First of all, the small animals, they tend to have a greater body surface area per unit of weight than the large animals. So therefore, they tend to have relatively high weight specific rate of water loss. Second reason might be that small animals, they tend to have a higher metabolic rate per unit of body weight than the large animals. Therefore, they tend to have high weight specific rate of respiratory water loss. There might be also some differences among the phylogenetic groups in their total rate of evaporative water loss. So let's move on to the terrestrial vertebrates that are living freely in their natural habitats. So terrestrial animals, both humidic and xeric, enter the dormancy, often called the estivation, in response to immediate predictable water stress. So mammals and birds that enter the water-related dormancy, including the desert ground squirrels and poor wills, they often undergo metabolic depression and become hypothermic. So metabolism is depressed during dormancy in poikilotherms as well. So this metabolic depression has several advantages. The first advantage is that An animal in metabolic depression can live on body fat or other stored food for a long time. Second advantage of this is that animals' requirement for oxygen is reduced, therefore reducing its respiratory evaporation. And finally, the third advantage is that nitrogenous and other wastes are produced at a lower rate, which may be vital in permitting the protected existence with little or no urine output. So here in this graph, you can see the total rates of water turnover of free-living terrestrial animals. They follow the allometric patterns. So this means that here in this figure, it shows the general water turnover pattern of vertebrates living in the wild. So since mammals and birds of any given body size turn over far more water per day than the lizards, snakes, or other non-avian reptiles of the same size. So there is a particular formula for that. The total, the T is equal to AW. So where T, it represents the total rate of water turn, T tends to be an allometric function. So exponent B, which is typically 0.6 to 0.8. This means that if we consider the relation between weight specific rate of water turnover and weight, the exponent is negative. So animals that live on land, we have a case study. So we will now look at the water relation of some particular groups of animals on land. Doing so will provide an opportunity to integrate the points that are made in the previous sections. So amphibians occupy diverse habitats despite their meager physiological abilities to limit this water loss. We have certain terrestrial amphibians that provide an instructive case study because they have invaded an impressive variety of habitats from the shores of ponds to deserts. So this diversity of habitats, most species are humidic animals, regardless of where they live. They are remarkably similar to one another in their physiological water balance characteristics. So how do dehydration-prone amphibians? They live in deserts. So as already mentioned, some species of frogs and toads, such as we have the Bufo cognatus and the spadefoot, that's the Scaphiopas coca in North America. They have skin that provides no more protection against the evaporate water loss than those of semi-aquatic frogs, such as we have the leopard frogs, like here you can see in the picture. And then they live successfully in deserts or other arid habitats. So such desert species are in fact remarkably similar to the majority of terrestrial amphibians in all physiological respects. So you can see here in this figure, so frogs of this sort have two genera. One is a Phylomedusa that belongs to South and Central America, and other is the Chiromantis of Africa that have been studied for several decades. So the lipids are sometimes are spread on the outside of skin rather than being incorporated within the skin tissue. So Phylomedusa, that's one of the genera, for example, it excretes the lipids from skin glands and spreads them on its skin surface. A second highly distinctive trait of the genera is that they excrete much of their nitrogenous wastes as a poorly soluble uric acid, or we can say urates. For example, we have the Phylomedusa savagi, that's the another species that excretes much of its nitrogenous wastes as a poorly soluble uric acid. So talking about the diurnal desert ants, so certain insects are among the most successful of all animals in severe desert conditions. So being successful, they provide succulent food for other less adept desert dwellers. So one of the most interesting phenomena in the living world is presented by desert ants that feed on other desert insects killed by heat. So life for this diurnal insect in severe deserts is so tireless that every day some individual accidentally die of overheating despite extraordinary adaptations for desert existence. So desert ants of several species, they scavenge the bodies of such heat-killed insects. So the deaths that often occur in the heat of the day, thus to get moisture from their prey, the ants must venture forth from underground burrows in the heat the day to gather the dead bodies of those animals before the sun quickly bakes the bodies dry. So species of such ants in deserts around the world have evolved unusual long legs, as you can see here in this figure. So these long legs, this is believed to be related to the fact that air temperature declines extremely steeply with altitude above the sun heated. So these stilt-like legs, although they elevate an ant's body just millimeters higher than it would be otherwise, so they can reduce the air temperature to which the body is exposed by about 10 degrees centigrade. So these still like legs, they actually protect them against this temperature. So the ants also exploit the steep temperature gradient at times by taking breaks from desert foraging to climb up on pebbles or other high points in the desert to reach even lower air temperature than their stilt-like legs they permit them. So this was about the diurnal deserts, that how they collect the heat-killed insects and their the stilt-like eggs that keep them above the intensely hot sand surface. So talking about uh, these kangaroo that live in the desert, so we can say that studies of lizards and small mammals, they help clarify the complexity of these desert existence. So we have certain kinds of species of small desert mammals that by far have attracted the most curiosity. So like here in this figure, you can see the kangaroo rats. These contribute to the genesis of modern animal physiology, and the researchers, they have produced a water balance summary for this kangaroo rats. So they had no drinking water, and animals were studied at 25 degrees centigrade. So when this research was carried out, or we can say for this study, when that was carried out, the actual temperature was like 25 degrees centigrade for these rats, and they were fed mostly the barley grains at equilibrium with atmospheric moisture, and they have not been provided any drinking water. So usually they do not drink even if water is available. So water loss is that's shown in the red lines, or the minimum possible water loss. In actually, if water intake exceeds minimum loss, the animals increase their losses by excreting more dilute urine so that the losses match the inputs. Inputs are here in the blue line. So this red line here, it denotes the total water loss. So amounts of water graphed on the y-axis are those gained or lost each time 100 gram of barley is consumed. Under the conditions of study, the animals normally consume about 100 grams of barley in about a month. So this graph here, it shows an index of water balance. That's the metabolic water production as a ratio of total evaporative water loss, or we can say EWL. So this ratio provides a useful index of ability to stay in water balance for species in which metabolic water production and evaporation are the principal processes of water gain and loss. So shown in this figure here, we have the data for two populations of kangaroo rats. That is the Dipodomys miriami and two species of desert birds, the dune lark and zebra finch, that's called as Porphyra gutata. So these are the different, this is the data for these kinds of animals. So moving on to the lark species. So water availability in a species habitat is quantified by an index that can vary from like 1.5 to 3.5. 1.5 is when there is very arid conditions and 3.5 when the conditions are moist. So in each graph, each symbol represents a different species of lark. So metabolic rates and rate of water loss are expressed using indices that are designed to be independent of body weight. So metabolic rates shown are the basal metabolic rates that are in the red lines, uh, and average free living metabolic rates that denote the black lines. So rates of water loss shown are rates of water evaporate to water loss, that's the red, and free living water turnover rates, that's the black. So activity is actually the percentage of daylight hours that's spent active. So that was all about the water and salt physiology of animals. So moving on to the next chapter, we will be talking about the kidneys and the excretion. So let's start with the next chapter. We will be talking about the kidneys and excretion. That means we will see in this chapter that kidneys of various types of animals, they are very diverse in morphology as well as in details of their physiology. So all kidneys, however, they have uh, features in common. For example, they have three features in common. First, they all consist of tubular elements that discharge directly or indirectly to the outside world. Second, they all produce and eliminate aqueous solutions derived from blood plasma or other extracellular body fluids. And third, their function is the regulation of composition and volume of blood plasma and other extracellular body fluids by means of controlled excretion of solutes and water. So let's take an example of species of small mammals that have evolved in deserts or among the all animals that extreme performers in their ability to concentrate their urine, with which the kidneys are mostly involved. So excretion of dissolved wastes in as little water as possible is a key to maintain the water balance in water poor environment. For example, in deserts. So that's one of the ways that it might be a key to maintain this water balance in this water poor environment. So around the world, each desert has its own assemblage of small mammals. Some of these mammals, such as the kangaroo rats that you can see here in this figure, these are the kangaroo rats of the American Southwest. They have been carefully studied. So others, such as the gerbils from Middle East deserts, have been little studied by physiologists. So shown here are the Miriam's kangaroo rat, that's the Dipodamus miriami from the American Southwest, and the four-toed gerfog, that's from the Northern Sahara in Egypt, Libya. So animals' body fluids are dynamic. We all know that they continuously gain and lose water. So inorganic ions and organic fluids. So this is continuously going on. So the kangaroo rat in the desert is successfully maintaining water balance in moderate summer weather. About 15% of the water in its body fluid is lost and replaced each day. So this rate of turnover is low for a mammal of its body size. So the mice and rats of moist uh, temperate habitats often turn over like 35% or more of their water per day. So humans, we all know that being larger, they have lower percentage of turnover rates. But still, when the people go about their ordinary daily lives, about 7% of the body water is lost and replaced every day. So inorganic ions and organic solutes in the body fluids, they also undergo some insensitive turnover. So great dynamism of body fluids means that their composition is continuously in danger because shifted away from normal. So as water, for example, leaves the body fluid and is replaced each day, any mismatch between the rate of loss and replacement occurs, which causes the body fluid to become too dilute or too concentrated. So kangaroo rats and other desert rodents have been studied mostly in the physiology of kidney functions, not unlike the status of cheetahs in the physiology of running. So kidneys of these desert rodents present the ultimate product of evolution in their ability to concentrate the urine. So most types of animals, they cannot concentrate their urine at all. So that's most cannot produce urine with an osmotic pressure higher than the osmotic pressure of their blood plasma. So what actually urine is? Urine is the product of kidneys that's typically a complex solution containing multiple inorganic and organic solutes. So all the constituents of the urine, including water, they are drawn from the blood plasma, and the urine concentration of each affects the blood concentration according to the principles we already discussed. So here in this figure, you can see the structural and functional basis for the formation of primary urine by ultrafiltration in the vertebrate kidneys. So we all know that urine formation can be conceptualized as occurring with two steps. So first is an aqueous solution called the primary urine is introduced into the kidney tubules. And second, this solution is modified as it moves through the kidney tubules and then other excretory passages. So ultimately it becomes the definitive urine that's eliminated. So these are the major two functions. The first, an aqueous solution is formed as the primary urine, and then this passes through different kidney tubules and other excretory passages. Ultimately, it becomes the urine. So there is a process that occurs in this in the kidneys, that's the ultrafiltration. So ultrafiltration is the process that occurs. So the process is a form of filtration that because the solutes of large molecular size typically are unable to pass through the wall of the tubules. So thus the fluid introduced into the tubule lumen, which is termed as filtrate or ultrafiltrate, consists only of water and subset of solutes that are able to stream through within the water. So blood plasma is the source of the water and the solutes that stream through. So there are exceptions in some groups of invertebrates where the blood pressure produced by the heart is typically the pressure that drives this ultrafiltration. So to understand well the formation of primary urine by ultrafiltration, let's examine the process in the vertebrate kidneys. So here in this figure, you can see the process that mostly exemplifies, or we can examine this process of ultrafiltration in the vertebrate kidneys. So each kidney consists of many tubules, which are called as nephrons, the walls of which these nephrons consist of a single layer of epithelial cells, like here you can see in the figure B. So each nephron begins with this blindly within its walls through into a hemispherical invaginated structure, which is called the Bowman's capsule, named after the William Bowman, which was a scientist who first described it. So tucked inside each Bowman capsule is an anastomizing cluster of blood capillaries. These are termed as glomerulus. So this is the wall of the Bowman's capsule. So Bowman's capsule and glomerulus together constitute a renal corpuscle. The glomerular capillaries are intimately, they are tucked to the inner wall of Bowman's capsule. So these intervening structures, they have a specialized morphology and they act as filter. So fluid is driven through this filter from the blood plasma into the lumen of the Bowman's capsule by the hydrostatic pressure of the blood. The fluid that accumulates in the lumen of the Bowman's capsule is known as the capsular fluid and is the primary urine. So our first step is completed here. So we got the primary urine. So all the critical determinant of whether a fluid will pass through its molecular size, molecular charge, and shape can also be significant. Thus, the filter has complex features which include, but they are not limited to these physical pores or slits. Inorganic ions, the small organic molecules such as glucose, urea, and amino acids, they move freely with filtered fluid as it passes through blood plasma into the lumen of Bowman's capsule. So because the proteins, they remain more concentrated in the blood plasma than in the capsular fluid, the osmotic pressure of blood plasma is higher than the osmotic pressure of capsular fluid. So moving on to the figure C here, that focuses, that forces that, or we can force it that favor and impede the filtration. So how this occurs is that blood pressure in the glomerular capillaries is significantly higher than the blood pressure in most capillaries in mammals. So this helps to promote the filtration and formation of the primary urine. So part of the reason for the high capillary blood pressure is that arterioles, they lead to the glomeruli are relatively large in diameter and thus they offer relatively low resistance as blood flows to the glomeruli. So the rate of primary urine formation by all of an animal's kidney tubules taken together is called the filtration rate. So in vertebrates, it's termed specifically as glomerular filtration rate or GFR. So let's move on to the next slide. We will talk about the formation of primary urine by active solute secretion. So to see how the urine formation can be initiated by secretion, let's consider this figure, which represents the essentials of a secretory system in a stepwise fashion. So the first step in this process is the osmotic pressure and the concentrations of both solutes are equal on the inside and outside of kidney tubule. So this is the step first. So the osmotic pressure is same, both outside and inside the kidney tubules. The blue dots here, they denote the solutes X and the red dots denote the solute Y. So moving on to the next step is here, the energy from an active transport pump uses energy from ATP to secrete a quantity of solute X, which is shown with the blue dots, into the lumen of the kidney tubule, increasing the inside concentration of X and also increasing the inside osmotic pressure. So most of the blue solutes, they move inside, so they change the osmotic pressure of the solutes. So in step 3, the water moves inward. So you can say that water in step 3, the water also enters the tubular fluid by osmosis, diluting this solute Y, because Y is then diluted inside. And then finally, the fourth step. So because of this, what happens when the water moves inward by osmosis following the osmotic gradient that was set up by the secretion of solute X and the volume of fluid in the tubule, it increases. So because of this increase in volume, the inside concentration of solute Y, which was initially same as the outside concentration, is reduced. So that's not now lower than the outside concentration. The final step, which is the step 4, the solute Y diffuses inward following its concentration gradient. So we can say that active secretion of even just a single solute into a kidney tubule can lead to a passive influx of water in other solutes. So thus a complex solution of many solutes can be introduced into the lumen of a kidney tubule from the body fluids, so bathing the tubule by a secretory mechanism. So the permeability of epithelium to the various solutes that might passively diffuse into the tubular lumen, it determines which solutes do in fact enter. So let's move on to the amphibian nephrons and their connections to collecting ducts. So how this works? The amphibians, we all know that they provide an excellent starting point for the study of vertebrate nephron function. So there is not much known about the amphibian nephron because of practical considerations that make up the nephrons of amphibians easy to study. Furthermore, the amphibian nephron can reasonably be considered as generalized vertebrate nephrons. So our main purpose in this section is not only to describe how the amphibians form urine, but also to bring out many additional general principles of vertebrate kidney function by examples. So like here in this figure, you can see that each nephron of an amphibian consists of a Bowman's capsule, a convoluted segment known as the proximal convoluted tubule, and then we have the intermediate segment, we have the distal convoluted tubules, and then finally we have the collecting tubule, which is here. So you can see in the figure A here, which unfolds amphibian nephron, and figure B, we have a nephron with the this toad bufo that's shown realistically in its natural configuration. So we have a C, that's the single collecting duct showing the connections of the collecting tubules of many nephrons also. So these actually, we can say that there are hundreds or thousands of nephrons that are found in each kidney, and the nephrons constitute much of the bulk of kidney tissue. So in each nephron, the structure and function of nephron epithelium change along the length of the nephron from one segment to the next major segment. So structure and function, they also often change within each major nephron segment, for example, along the length of distal convoluted tubule. So in each kidney, the collecting tubules of all the nephron feed into the collecting duct. We can see the figure C here. So this is the collecting duct showing the attachment of many nephrons. And all the collecting ducts connect to a single ureter which carries the fluid from the kidney to the bladder. So this is the ureter which throws away this urine to the bladder. So we have certain major solutes and water fluxes in the late distal convoluted tubule during the diuresis. So we can say that extent of pure water excretion is actually controlled in the distal convoluted tubule by varying the degree to which osmotic water reabsorption keeps pace. So the extent of water reabsorption is actually controlled by modulating the permeability of walls. So control of permeability is actually exercised partly by the ADH, which means the antidiuretic hormone, which is secreted by the neurohypophysis. That's the posterior pituitary gland. So as we now explain these points, let's refer to the figure uh, in this slide for a visual summary. So when at ADH levels, that's the antidiuretic hormone levels, are low, we can see the figure A here. So when this antidiuretic hormone is low, what happens is that the walls of the distal convoluted tubule has a low permeability to water. So consequently, we can say the sodium chloride reabsorption and water reabsorption from the tubular fluid, they are significantly uncoupled. So active reabsorption of NaCl tends to dilute the tubular fluid and thus create an osmotic gradient. Moving on to the next section, that's the B, that's when the antidiuretic hormone is high. The presence of ADH induces the wall of the late distal convolute tubule to become relatively permeable to water, and the distal tubule then functions more like the proximal tubule. So this osmotic water reabsorption is promoted, thus NaCl, or we can say sodium chloride, is reabsorbed. More water is reabsorbed than when antidiuretic hormone levels are low. So this ADH, actually, the antidiuretic hormone, is believed to control the water permeability of amphibian distal convoluted tubule by controlling the insertion and retrieval of aquaporin proteins. So what happens is that when the level of this antidiuretic hormone is high, the aquaporins are inserted into the cell membranes within the water channels, therefore in place in cell membranes. So water can pass easily through the epithelium relatively uh, readily by the osmosis. And when the level of ADH is low, these aquaporins are retrieved from the cell membranes. That's they are returned to intracellular locations. And osmosis through the epithelium is stopped. So more will be said of aquaporin function. So let's now talk about the evolutionary development of renal papillae in mammals that are native to different habitats. So even before the physiology of the loops of Henle began to understood, morphological evidence strongly suggests that loops are intimately involved in the production of urine. That's the hyperosmotic into the blood plasma. So another type of comparative morphological evidence comes from studies of these renal papillae that you see here in this figure. So not all the mammals have a grossly apparelled renal papillae. Commonly, however, the renal medulla has a roughly pyramidal shape and forms a projection into the lumen of the renal pelvis. So like here in this figure B, you can see the papilla was more evident in species from the mesic, that's the moderately moist habitats, and was most developed in species from arid habitats. So inspired by the Sperber's work, there was a scientist, his results, they indicate that there is a greater evolutionary development of long loops of Henle in species that produce relatively concentrated urine. So, or we can say the thickness of the medulla provides a measure of lengths of the longest loop of Henle. So moving on to the next slide, that's the figure here. It shows that a high relative medullary thickness means that the longest loop of Henle or long relative to the overall dimension of the kidney. So this here, these graphs, they give you maximum urine concentration that correlates with the relative thickness of the medulla. For example, the relation is plotted for 68 species of mammals. So each point represents a different species. So relative medullary thickness to a dimensionless number. To calculate it, an index of kidney size is first calculated by taking the cube root of the product of three principal linear dimensions. That's the length, width, and thickness of the kidney. So medullary thickness is then expressed as a ratio of index of kidney size to obtain relative medullary thickness. So this graph here shows the relation between the relative medullary thickness and body size that depends on whether mammals are native to arid, mesic, or aquatic habitats here. So you can see it gives us, so this is actually the work that was done by Sperber's scientist, his research, they were, so he gives us the latest incarnation of this work that shows in this, that's shown in this figure, where relative medullary thickness is plotted as a function of body weight, or we can say the size for mammals from these uh, three types of habitats. That's the mesic, native, or aquatic habitats. So habitat, however, has a significant effect, or it's a significant factor. So we can say that both the body size, just as concentrating ability tends to decrease with these body size, as you can see in the figure. So habitat, however, has a significant factor at any given body size. Mammals from arid habitats, they tend to have the thickest medullas and longest hoops of Henle, whereas those from freshwater aquatic habitats, they have the thinnest and shortest. And those from the intermediate, or we can say mesic habitats, they are in between these. So here in this figure, it gives a dramatic morphological comparison of kidneys of three species of rodents of similar body size. You can see that two of the species, that's the Mongolian gerbil and sand rat, evolved in deserts. Both have far more prominent renal papillae and thicker medullas than the laboratory rats. So moreover, the sand rat has a thicker, longer papillae than the gerbil. So detailed studies of the sand rat reveal that its renal medulla is elaborately organized in comparison with most mammals that are especially high proportion of long loops of Henle. So moving on to the generation of the single effect in the loop of Henle, how it occurs. We know that the cells in the walls of ascending thick segment of loop of Henle, they actively transport the sodium chloride from tubular fluid inside the loop into the medullary interstitial fluid. So consequence of this NaCl transport, as you can see here in this figure, it depends on the permeability characteristic of the ascending limb and the adjacent descending limb of the loop of Henle. So walls of the ascending limb are essentially impermeable to water. So in a few words, we can say that active transport of this sodium chloride out of the ascending limb, it lowers the NaCl concentration and osmotic pressure of the ascending limb fluid. And it raises the NaCl concentration and osmotic pressure of both adjacent interstitial fluid and adjacent descending limb. So these differences between the ascending limb fluid and the adjacent interstitial and descending limb fluid, it represents the single effect. So this we call as the single effect. So here in this figure, you can see this is the single effect. That's the difference in osmotic pressure and sodium chloride concentration between the ascending fluid and adjacent interstitial fluid and descending limb fluid. So here are some of the major molecular mechanisms of sodium chloride reabsorption and associated processes in three parts of the mammalian, this kidney tubule. Some studies of fine structure of papilla of the medulla in young laboratory rats, it actually shows the modern molecular and genomic methods, they are enabling the kidney researchers to study subjects that seemed utterly beyond reach 25 years ago. So when this is the case, elements can be distinguished by immunological labels. So for immunohistochemical study in this figure, the fluorescent antibodies were prepared against three distinguished proteins. We have a blue fluorescing antibody that was prepared against aquaporin, and then we have red and green fluorescing antibodies that were prepared against aquaporin 1 and the urea transporter that was found respectively. So similar kinds of antennal glands and the urine formation in a freshwater crayfish. Uh, we know that each antennal gland is a single tube, sometimes loosely described as a single giant nephron. So in a freshwater crayfish, you can see each antennal gland begins with a closed end sac, or you can say the ciliomosaic that's lying to the side of the esophagus. And also uh, in the in this figure, you can see that there are the walls of the end sac. They are thin, and arteries from the heart. They supply a network of small vessels, or we can say the lacunae on the outer surface of the end sac. So this morphological evidence has suggested that fluid enters the end sac by filtering under the force of blood pressure. So how the urine formation occurs in mollusks is that the renal organs of mollusks, they are tubular or cellular structures, which are called nephridia or kidneys. So most thoroughly understood, the molluscan kidney is of a giant octopus, or we can say Enterooctopus doflini, an ocean mollusk. So here in this figure, you can see the kidneys of octopuses and squids. They are associated with the bronchial hearts, that's the gills, rather than the systematic heart. So each bronchial heart bears a thin-walled protuberance, which is called the bronchial heart appendage, which communicates with the lumen of the heart. So moving on to the next slide, which gives us the average composition of the rectal fluid and other body fluids in locusts that were deprived of food and provided with either tap water or saline solution to drink. So here are the rectal experimental treatment was the water fed or saline fed. So for example, we have this the fasting locusts. They were permitted to drink either tap water or saline solution containing the potassium, sodium, and chloride and other ions. So rectum in the water-fed locusts, it reclaimed the ions, returning them to blood. So, however, rectum in the saline-fed locusts, it raised the ion concentration in the rectal fluid with the anterior hindgut fluid. So talking about the structure and function of blowfly rectal papillae, each blowfly has 4 rectal papillae. So in the figure A, you can see the structure of papillae that's high dramatically the intercellular spaces that are depicted for as a single broad cavity running through the epithelial cells actually consists of a complex interconnecting network of small channels. And you can see here. And then we have the figure B that proposed the process of water absorption from the rectal lumen. So blue arrows here, they denote the water flux, and the red ones, they denote the solute flux. So some nitrogenous compounds, they are excreted by the animals. So some of the compounds in which animals incorporate nitrogen during catabolism, they are shown in this figure. So each of these nitrogenous end products has advantages and disadvantages. For example, some of the compounds, they are relatively cheap to make, whereas others are low in toxicity. So there is no single end product that is ideal in all ways. Moreover, there sometimes whereas end products often represent wastes to be excreted. So they sometimes can be used in adaptive ways. So with all these considerations in mind, we cannot be surprised that animals have evolved a variety of strategies for dealing with the nitrogen atoms that are released from organic molecules by the catabolism. So talking about the urea that's highly soluble and generally diffuse readily across membranes. So if urea is less toxic than ammonia, why so many animals, they are ammonotelic? That means, for example, bullfrogs, they shift from ammonotelism to ureotelism as they undergo the metamorphosis. So these American bullfrogs, they until recently named the Rana castiviana, which started life as an aquatic tadpole. So metamorphosis occurs, and then developmental biologists, they identify the stages of development using the stage numbers on the x-axis. So activities in the liver of four enzymes of metabolic pathway that synthesizes urea are shown as a function of the stage. So excretion of urea, which is also shown in the red line uh, here, is now recognized to represent the activity of two enzymes that together produce the arginine. So there is a persuasive evidence that vertebrates adopted ureotelism when they emerged into land. So one line of supportive evidence is that certain amphibians are usually amertelic. So they express the enzymes for ureosynthesis increasingly as they go through the metamorphosis and eventually become ureotelic adults. Okay, so that was all about our chapter second. That was about the kidneys and excretion. Let's move on to the third chapter. We will be talking about the animals in the dry savannas. So how the water, salts, and excretion takes place in deserts and dry savannas. So we will be talking. So water, salts, and excretion in mammals of deserts and dry savannas, how they actually interact with the high temperature of the deserts. So in deserts, there are large herbivores such as oryx. You can see in this photograph here. They can survive indefinitely in hot deserts without drinking water. They are usually the equivalent of nomads moving about to find moisture to survive. So similarly in the dry savannas, the grassland plains, large herbivores often function as nomads or migrants. So conditions might vary too much from time to time and from place to place in these arid zones that living in an invariant locale is often not a viable strategy. So our goal in this chapter is to explore how the oryxes and other species of mammals, for example, 5 kg or larger in body weight, can live in hot deserts or dry savanna ecosystem. So one way to understand these mammals is to compare them with small species. As we already mentioned, the kangaroo rats, the gerbils, and other small mammals that coexist in arid habitats. And however, the small mammals we already discussed in the previous chapter, and we will not turn to them here in any detail. So mammals of Africa will receive the greatest emphasis because they form the most diverse and abundant set of large arid land mammals. So deserts and semi-deserts, by most accountings, they cover about 1/3 of the total land on Earth. So there is no uncomplicated way to define a desert. So one of the simplest definition is that a desert receives less than about 25 centimeter, or we can say 10 inches of precipitation per year. But this is an imperfect standard because there are regions which receive more than that, but they are undoubtedly the deserts. So following the lead of Emmanuel Noy, we can say that biologists, they prefer a definition that emphasizes two attributes. First, a desert is a place where precipitation is low and availability of water exerts a dominant controlling effect. So why are the features of the what are the different features of the dry savannas? So dry savannas, these are actually endless plains of the grass with trees dotted here and there. So taking a big picture, dry savannas are in many cases the neighboring environments to the deserts. So deserts and dry savannas, they exist for several reasons. So single most important cause of desert and semi-desert conditions are whereby air warmed at the equator, it rises to high altitudes, and then it displaces that air at those altitudes in such a way that high altitude air descends to Earth's surface. So talking about the relations of animals to water, so large animals, we all know that they have both disadvantages and advantages compared with the small mammals in hot deserts and dry savannas. So many of the most apparent disadvantages of the large size or the behavioral. So large animals, they cannot take shelter underground by digging burrows as small rodents do to protect themselves from the high temperatures, and they are less likely than small species to find adequate shade or protective microhabitats on the surface of the ground. So in hot environment, large animals therefore confront the heat more directly than the small mammals. So large body size might be a physiological advantage in terms of water costs. So to explore the physiological implications of body size, let's compare mammals of large and small size. So we can say that there was a scientist whose name was D.B. Dill. So he in 1930s, pioneer in the study of exercise and heat. He undertook famous walks in the desert that provided the very first quantitative information on water costs of mammalian exercise under hot conditions. So he himself was one of the human subjects, and he took along with him both dogs and burros. So together, man and beast, they trekked under the searing sun in the Nevada desert. So we will now discuss how he related his findings. We'll focus on the red and blue lines in this figure. Both lines show the rates at which animals must lose water by evaporation to thermoregulate under hot conditions as a function of body size. So the red line was constructed by application of fundamental principles to Dill's original data, and later Dill and collaborators, they gathered more data of smaller sort or burrows and high school students in the Colorado district. So the red line shows the very strong relationship between body size and the water cost of thermoregulation when mammals are active in the daytime desert. So as you can see here, the water cost of thermoregulation is predicted to soar extraordinarily high levels of small body sizes. So the blue lines, like I said, it denotes the blue line and blue point. They represent the humans and animals that are resting at high air temperatures. So talking about the coexisting species that are diverse in their relation to drinking water, you can see here that one might imagine that all large species of mammals have long evolutionary histories. So among the species that are common in dry, hot places, some are classified as drinking water independent, and some are classified as drinking water dependent. So drinking water independent, why they are named? Because they are able to remain healthy for many days or weeks without access to drinking water. And drinking water dependent means that coexisting species must drink each day or at least every day. So that's why these are classified as drinking water dependent. So in this figure, you can see here the grants, gazelles on such days, they are indifferent to sun or shade. So this contrast in shade-seeking behavior, it correlates with the water physiology of two species. The species that dehydrates more rapidly in the absence of drinking water. The most dramatic manifestation of the difference between drinking water independent and drinking water dependent species is the Serengeti ecosystem, which is the differential participation of the two categories of animals in the animal migration. So these grand gazelles are drinking water independent antelopes that are noted for their indifference to sun or shade. So most abundant migratory species is the wild beast, which in recent times has numbered about 1 million. So another highly visible migratory species is the zebras. So both the wild beast and zebra, they are drinking water dependent species. So they spend the dry season in far northwest, the wettest part of ecosystem. So then as tiny season starts in November or December, they move on a broad front from northwest towards southeastern plains. Like here you can see. So this is the annual migration in the Serengeti ecosystem, a dry savanna ecosystem. So the table here, it shows the water use by people in 4 parts of communities in Arabia. So these are the modern Arabian town without major industry, and then we have traditional agricultural village, we have small desert settlement, and then with the supply by government water truck, and then we have small desert settlement with traditional water supply. So this here, the table, it shows the uh, indigenous people survived on very small amounts of water compared with modern urban water usage, and they obtained the water they needed by moving with the rains. So the next slide here, it shows the moisture content of the dry grass varies with the time of day or night. So water and food resources actually in the deserts and dry savannas, they are very complex. So here in this figure, it shows the data that was gathered on dead brown blades of the perennial desert grass in the Nam Desert. So water content of grass is highest near dawn when the relative humidity is highest. You can see here. So the bar here, it shows the animal gets 2.7 times more water from parked grass if it eats at dawn rather than in the evening. So nutritional content of some plants varies with the rainy and dry seasons. So percentage we have for the large desert, for the large desert ecosystem, for example, they vary more in their protein content between wet and dry conditions. So a recent study of grasses in Arabian desert showed that protein content uh, of the computer related to plant dry weight falls exponentially during the long periods of drought from about 9% in the early weeks of drought. So after long periods of drought, the concentration of phosphorus, copper, and other minerals are sometimes too low in desert plants to meet the nutritional needs of these herbivores. So coming on to the next slide, we'll talk about the desert beetle. So when fog rolls in from the sea, as it does after often because of local conditions, the beetle goes to the crest of sand dunes and position itself as shown in a fog basking stance in which its broad dorsal surface faces into the fog-laden sea breeze. So as the fog water accumulates on the beetle's body, the water runs down to the beetle's mouth. So for the large desert herbivores that are our focus in this chapter, the effects of fog are more indirect. So fogs provide water to the plants that these herbivores eat. So this input of water not only increases the water content of plants, but it helps to maintain the plant health. For example, plant protein is higher than it's otherwise, making the plants more nutritious. So moving on to the dramatic adaptations of particular species, for the most part, the physiologists, they made the effort to study large mammalian of deserts, mammals of the deserts and dry savannas. So water balance physiologists tend to attach the greatest importance to adaptations that permit animals to live with as little water as possible. So with water balance physiologists, they organize most physiological research in the deserts and dry savannas. So oryxes, they represent the pinnacle of desert survival. So among the truly wild large animals that are the oryxes, they represent the pinnacle of evolution in their ability then to survive in the deserts. So dromedaries might be their equal physiologically, but wild dromedaries went extinct before historical times. So Arabian oryx is a species that weighs 70 to 100 kg, was driven to extinction in the wild for a long, but has been re-established. So these oryxes, they can live in some of the most inhospitable places on the earth. So moving on to next slide, which talks about the average body core temperatures of free-living Arabian oryxes over the 24-hour day. For example, here you can see that in this graph, it shows the 6 oryxes they were studied while living in their natural environment in Saudi Arabia and monitored using the implanted radio emitters during the summer and during winter. So this means that Arabian oryxes, they exhibit high amplitude cycles of body temperature in summer. So dromedary camels, they doesn't store water but conserve it and tolerate profound dehydration. So modern era of physiological research on large desert mammals began with studies on dromedary camels. So Nielsen in the mid 20th century, he steadily been learned since then. So dromedaries drink in part of because they are domesticated and human handlers water them. So from the time of ancient Rome, we can say that there are two principal keys to extraordinary ability of camels to survive in the deserts. Camels conserve water extraordinarily well. And also, based on studies of pet animals, we know that dromedaries dramatically exploit the water conservation benefits. So when dehydrated, they sometimes allow their body temperatures to rise as much as 6 degrees centigrade during the day and fall to the same extent in the cool of night. One reason they can permit their body temperature to rise as high is that they keep their brain cooler than the rest of their body. So a trait also reported in oryxes and gazelles. So these dromedaries, they employ several other water conservation strategies. For example, they can produce dry feces and concentrated urine. The other reason might be that they curtail their urine production. And third reason is that they have thick, sometimes glossy fur that acts as a heat shield. So second known key to the ability of camels to survive for weeks without drinking water is that their unusual tolerance to dehydration. So species of mammals without long evolutionary histories in deserts, such as dogs or horses, are in danger of death if they dehydrate. So these dromedaries, in contrast, can tolerate at least twice this extent of dehydration. So that was all about the different kinds of mammals that adapt to the high temperatures in dry savannas or deserts. Thank you so much for your time.